So we're going to wait just a second. Martha is going to say a few words in a minute, but I'll go ahead and get us started. I'm Christy Pattengill-Semmens. I'm one of REEF's executive directors, and I'm really excited to welcome y'all here. Hopefully you've had a great second day, I guess. It's the second day of REEF Fest, and we really had a lot of fun last night at the REEF campus for all of you who were able to join us. It was a really nice event. Felt really good to see people gathering again and kind of the energy and the excitement. It was really nice. So we're super excited that we were able to bring this together. We've said it before. It's, REEF is like a family, and it's nice to be able to bring that family back together. We appreciate everybody's thoughtfulness about the health and safety issues, wearing your masks indoors at all times. Inside the government center, inside here, they do not want any food or drink in here. So please be respectful of that. We'll have the hospitality set up outside after the seminar. Again, during the happy hour, we'll bring out some more food. For those of you who didn't find it, there's bathrooms out in the lobby. So feel free, it's casual. So if you need to use the restrooms at any time, go ahead. And what are the other? Oh, yeah, just in here, it's a big room. So, spread out a little bit. It seems like everybody's kind of seated with their family pods or their friend pods. So that's great. And just let's use the space. It's a big room to just maximize our health and safety. So I'm going to have Martha come up in just a second. And then I will make a couple more announcements, and then we'll get the seminar started.
All right, it's so great to see all of you again. And one person that I wanted to take a moment to thank, Leonardo, if you could come up. Leonardo works for Public Works, and he is the person that's helped make all of this possible. So he's been running around behind the scenes, moving tables, cleaning bathrooms, coordinating with all the people that work in this building. So we literally could not do it without all of your help. We had a gift that we wanted to give to you, Leonardo. Thanks so much for all your work that you've done this year. And this is what, the 5th year that you've done REEF Fest with us? The 5th year that you've done REEF Fest with us? 7th year. So every year, thank you so much. We really appreciate it.
We really wanted to.
Thank you very much.
Leo's been asking about that shirt every day this week because last year I forgot to give him one in 2019. So 2 years later, finally brought the shirt.
Yep. Yeah, in addition to Leo, up in our booth, we've got David Hartman. Also could not do it without David. Woohoo! David's been a longtime REEF member and volunteer, and he has helped make the magic happen with all of the AV. And this year, we've been wanting to live stream these talks for a long time for people who couldn't be here. And this year especially, there was people who just couldn't be here because of COVID. And so these are all being live streamed on Facebook and on YouTube. So all of our members, no matter where they are, can watch them as the magic happens and then be able to watch them later because they're all going to be archived. So if you miss any of the seminars this weekend, you'll be able to go and watch them there later. So we really appreciate that. David also has Daryl Duda helping kind of on the sidelines. He's the backup, the mic man. So thank you guys. I know it's a lot. You do it every time. So thank you. A couple other things. So like I said, right after this, we'll do happy hour right here in the building. So we're not going anywhere. About an hour and a half of just some light snacks and fun. And we'll have the bar open, silent auction, raffle tickets. And then we'll come back here for an amazing evening with our founders, Paul Heumann and Ned Deloach, and hear about their heady hunt for 5,000 fishes. That's going to be a great time. I really want to say thanks to all the donors and businesses who helped make that silent auction happen. So take a look at those. The sponsors are all in the program as well. First Horizon Foundation, they gave generous support of the event. The Monroe County Tourism Development Council, all those folks helped make this possible, as all you do as well. We couldn't have an event without people being here. So thank you for making the effort and the time to be here, and we hope you're really enjoying it. So Any opportunities to, bid, support REEF, we greatly appreciate your support. So without further ado, I'm really excited about this talk. It came together quite well, and it's going to be a fun one. So the idea, reefs conservation stories from the field. We have a lot of conservation science programs that we do. So when we're talking about reefs programs, the cornerstone, the citizen science program, the volunteer fish survey project. And out of that grew the Grouper Moon Project, which is a spawning aggregation, Nassau Grouper spawning aggregation research program. And then the invasive species, the lionfish showed up on the scene. And so we have a invasive species research program. And then of course, kind of scaffolded on all top of that is our education and outreach, which is a critical piece of the work that REEF does. And so we have a lot of fun stories to tell you from these different conservation programs through the years. And mostly we're going to talk about technology today, because there's definitely been some fun changes in technology and how we're making advances for marine conservation. So there's going to be 4 of us who are going to give this talk. So myself, I'm going to go at the end. And Brice Semmens and Dr. Brice Semmens, Dr. Scott Heppell, they're longtime reef science advisors. Brice actually also was one of the very first reef interns along with me. So that's where we met. And in '93, and then Scott's on our board and also serves as a really great mentor for our internship program. And then Dr. Alli Candelmo, she's also on REEF staff. She's our conservation science program manager. So the 4 of us are going to share a little journey through the years, through technology, and how REEF has leveraged those, sometimes successful, maybe sometimes not so successful, to advance our program. So without further ado, I will turn the mic over to Brice, and he's going to set the stage on this talk.
Okay, thank you. Hi, everybody. Can you hear me okay? Okay. Yeah, so like Christy said, we're going to talk a little bit about one particular focal area of our conservation stories through the years here at REEF. And that focal area revolves around one of the core tools that REEF uses, and that's conservation technology. 30 years. REEF's been around for 30 years. That's a long time. And in those 30 years, a lot changes. And, some of us are still really good looking and others of us not so much. And that's just kind of the way it goes. But when this started, shortly after that, Christy and I were interning, as she pointed out, with REEF over the summer. And even back then, we were making use of technology. And these are the pieces of technology that we were using at the time. On the left, that's the very, very fancy and very expensive, I might add. Those are $1991. Apple computer, I believe this was color at the time. Yeah, and it probably had whatever, a 12,000 baud modem or whatever it had. It was super high-end technology, right? And this was my machine here on the right. I bet none of you have ever seen that machine before. It was produced for about a year and then discontinued, but that is Apple's first Macintosh. So from these humble beginnings of computing technology, we were able to work over the summer to help further REEF's mission. And so over the course of this talk, we're going to be moving forward in time, marching over those 30 years, and we're going to be talking about technologies that we've made use of and how those have evolved over time. Speaking of technology, of course, the imaging technology has changed quite a bit as well, and I'll be talking about that. So on the left here is one of REEF's founders, Paul Heumann, using at the time very, very cutting edge imaging technology, right? There's no digital in that. Digital did not exist, of course, back then. This is slide film, so you had at most 36 exposures. You better make your shots count, right? And when I met Christy that summer, she had one of those. What is it? Who knows what that is on the right? An Iconus 5. High-end stuff, right? Why are they holding it out of the water? Because if you put it in the water, it floods. That's how that thing worked. Like literally every third time you used it, it was going to flood. Also slide film, right? So these were the two. And think about that. Like the original book, Paul and Ned's original book, was all from these machines. 36 exposures at most. It's pretty amazing to think about now because we take down cameras and we got the TG6, right? And that TG6 It's a waterproof camera that we put inside a waterproof case because we can make it as dummy proof as possible, and that's important for people like me. And that thing, you can fire off as many rounds as possible, right? And you can take really incredible pictures with zero actual talent. As witnessed by, this is an elegant plainfish that I took a picture of off of Costa Rica. Just, what was that, 2 months ago? Right, and so things have changed very, very, very significantly in those last 30 years. But of course, even from the beginning, REEF was as its cornerstone, as one of its pillars, using technologies. And of course, back then, high-end technology was scan forms, also a kind of imaging technology, and that's how we entered data. Why? Because the web had just been invented in 1991. There was no online data entry. That didn't come until much later, but we used scan forms. And that scan forms we continue to use up until what, 2007, right? And then some point along the way, REEF got a website, and now, of course, amazingly, we can enter in all of this data online, but REEF was very early in capitalizing on this. There are programs out there like eBird, run by the prestigious Cornell Bird Lab. This precedes that by a lot, way, way ahead of most of the other citizen science projects in terms of taking advantage of technologies. And REEF is reef.org. Like, think about that. Reef Sandals is not reef.org. That's a very hard URL to get. And why does REEF have that URL? It's because very early on, REEF recognized the value of the internet as a tool for communicating among citizen scientists. So at REEF's core always has been technology. And as that technology has evolved, REEF's continued to take advantage of that in different creative ways to make conservation science happen. And today we're going to talk about that. So this is just some of the many pictures of us using conservation technology in the field. Sometimes, Christy said, sometimes without great effect. That picture in the middle is us trying to fix something that didn't ultimately work very well, but sometimes that happens. All right, so we have, as Christy said, we've got a few of us talking. Dr. Alli Candelmo is going to first talk about collaborative innovation and the sounds of fishes, the songs of fishes, let's say. And then Dr. Scott Heppell is going to follow that up with a talk about satellites, microscopes, and the building blocks of all life on Earth. Heady stuff. I'm going to talk about imaging technology, and then finally, Dr. Christy Pattengill-Semmens is going to talk about efforts to bring emerging imaging tech to the citizen science world through some very cool AI stuff. Okay, so I'm going to stop talking. I'm going to turn it over to Alli.
Sorry, itchy mask face, which I'm not supposed to touch. Okay, so I'll be, as Brice said, talking a little bit about some collaborative invention with a team of scientists and nonprofits and government agencies, and then some acoustic research. So lionfish management has been, going on for decades now almost, but the deep reefs still pose a lot of issues. Removal of lionfish from these deep reefs has had some success using tech diving, rebreathers for spearfishing, but that's really limited in localized control. There's been a lot of robots developed, some more successful than others, some electrocuting robots that I haven't seen for a little while, but I did see in action initially, which was interesting. This Atlantic Share Reef Sweeper has actually been pretty successful in removing lionfish from deep reefs in Bermuda area. But overall, trap bycatch has actually been the most successful in targeting lionfish in those reefs 50 meters and greater. So there's some issues with traps, though, one of which is well known. Traps have a lot of bycatch. They catch a lot of fish, non-target species, mostly fish that go in there for shelter or food. Often they're baited. And then also the lobster traps that are baited for lobsters. If you're targeting that lionfish, you don't want to be catching this bycatch. You're trying to offset an ecological issue. We don't want to be killing native species that are ecologically important. What's the other issue you see in these trap pictures that may not make them not so effective for lionfish? Particularly the one on the right. They're not inside it. They use the habitat structure, but they don't necessarily always go inside. So you maybe have one in there, but is one lionfish enough to set, 100 traps at a time? So Dr. Steve Giddings, who's not here this week, but plays a major role in Grouper Moon Project and REEF in general, he's been working for decades now on a non-containment trap design that all of us have, been involved in one capacity or another through the years, because he brings that trap, or at least his little models, around with him everywhere, and loves to talk about it. But his goal was to create a transportable trap. You're looking at these, maybe not thinking transportable, easy to deploy and retrieve, but that they're going to minimize bycatch and have other, reduce all other ecological issues like ghost fishing, habitat damage along the way. So he's been kind of guiding this design through different iterations. We actually had this one out in Little Cayman with us when I worked out there. And then this one is a detachable FAD, which makes it a little bit more streamlined. FADs are fish aggregating devices that allow the lionfish to gather around them. But both of these proved to be too big, too cumbersome, bulky, difficult to retrieve and deploy. This one was good at keeping nurse sharks away from our tagged lionfish, and that's what we used it for in Little Cayman. So he has moved towards this more streamlined, foldable purse trap. And you can see the FAD is very narrow here, but that's really all lionfish need. They just need some structure and they'll aggregate towards it. And so by kind of using that flat FAD, the trap is actually able to close up. As it closes, you get this billowing effect that hopefully won't chase the lionfish away as it's closing, and they'll remain in the trap. And then there's minimal drag, less bottom impact. It hits those deflectors first and then opens sort of slowly, so there's not a big boom from a 70-pound trap. And then the retrieval is a bit more streamlined than some of the other designs. So REEF is now working through a grant funded by NOAA Saltonstall Kennedy program to determine if this Giddings trap, which is now patented, can be effective in catching lionfish. And so our goal is that this is the goal, to get that type of catch regularly, to make that trap cost effective and ecologically viable and economically viable. And so we will be working with scientists like Holden Harris and Alex Fogg and Steve Giddings that have been working with this trap in the Gulf of Mexico and various areas around the invaded region. But our main priority is to work with the Florida Keys lobster fishermen, because they know how to trap fish. They know how to deploy traps. They know where to put the traps. They know how long to put them down there. They know how to retrieve them. So we're collaborating with them as much as they will with us, setting up workshops, in-person meetings, to get better ideas of, how can we modify the design of the trap to make it fishable, to make it successful, and then make it able to catch lionfish. Yep. Yeah. So in Florida and throughout the invaded region, there's wholesalers that buy them, restaurants. It depends on the location, but in Florida, you can get upwards of $6.50 a pound for a whole lionfish. So there's been a small group of people that have monopolized, or not monopolized, but monetized on that commercial market. The lionfish that's all been through spear fishing in shallower reefs. The trap fishermen sell their lionfish bycatch, but when they go out for lobsters, but I'm kind of jumping ahead on this point, but they actually don't want the lionfish in the traps because they think the lionfish keep the lobsters out. It's really actually the reverse. FWC has been doing research with them, and the lobsters keep the lionfish out. So when they first deploy their set of traps, which is upwards of 500, there's 1 million lobster pots out in Florida waters. They get a big haul of lionfish, but after that, they bait the traps with lobsters, and so they don't get as much lionfish after that because there's already a lobster in there. So they don't make enough money. They make a lot of money on live lobsters, so they don't make enough money when it's lobster season to target lionfish, purposefully. And so the goal is to create a trap that they could use off season. Whether that is a modified lobster pot that eliminates bycatch and stops like most lobsters from going in and other fish species, or a trap like this. There's been, when we're speaking with the fishermen, I have to be, cautious to not say you're gonna have to use this trap. They don't want to use this trap. This trap has a lot of moving parts. They use a box with a big block of cement on the bottom. It's a lot easier, you know? So I'm just, you know, telling them, I'm just trying to gain your knowledge because there's places all over the region that don't let any trap fishing. So we want to learn how to fish this type of trap effectively in other areas. It may never be fished in the Florida Keys commercially, but maybe in other areas. But these are the guys that know how to do it. So that's the main goal as we're working with them. And some of the ideas we're looking to ask and find out are, you know, how much weight and buoyancy we need to attach to the trap, how to reduce drag resistance, you know, change the FAD design to be effective, how to make it open successfully, close fast enough so the lionfish don't come out, reduce any entanglement risks of marine species, and then whether to fish it like a trawl, like they do with their lobster pots where they're all attached, or single traps, which is much harder to go pick up single traps along the way. So the other groups that we're working with very closely is the FWC lobster research team down in Marathon. They've been working with lobster researchers, lobster fishermen, and also modifying the lobster pots for lionfish catch through the years. So they've been a huge asset. And then we've pulled in NOAA scientists, and in addition to Steve Giddings. So we just had a call with the NOAA gear research team up in Pascagoula, Mississippi. And I'll be sending a couple people with 2 traps to drive to Mississippi to try. I tried to ship them. Shipping those traps is apparently impossible unless you want to spend $5,000. So we're gonna have a couple of our interns are gonna have a road trip with some traps next week to bring them up to these guys because they're interested in tinkering with it and figuring out the different nuts and bolts and seeing if they can help make them as effective as possible. There's obviously REEF is a nonprofit. We're also working with Lionfish University and Reef Save to help promote the traps, and borrowed some traps with them initially. And then we work very closely with the local Florida Keys divers. They're particularly useful in helping us find sites that we can actually do the research on. Where can we find lionfish sites that we can deploy the traps and test out their effectiveness? So once we get the design that we want, we need to start deploying the traps, seeing if we can catch some lionfish. But one of the issues is if we just throw them in and pull them up without monitoring anything, we won't know why or if we caught lionfish, or and why we didn't catch lionfish. So we've been using technology to help support that research, running ROV cameras down with the traps to look at deployment success and retrieval success, and attaching cameras on the traps. And you can see just from the past few months, we've found out reasons why the trap may not open correctly. Sometimes it doesn't close right away, but it will close before it gets to the top. So at that point, all the lionfish would have gotten out. If we're not monitoring it with cameras, we'll just think there weren't lionfish there. So the ROV surveys are really important for that use. We're probably going to put a swivel on the trap so it stops spinning around like like that and making you dizzy in these videos. And then we can use those ROVs for habitat surveys as well, and looking for lionfish presence before and after the trap deployments. Along with that, we've got these programmable large underwater cameras that can record for upwards of a week that we can deploy with the traps and look at sort of habituation of the lionfish. How long does it take them to come there? How long do they stay? What time of day is the best time to pull the trap? Is the trap being dragged at all? Are there other species in the area interacting with lionfish? And then what's lionfish behavior can we learn just for ecological reasons and management purposes? We're going to be deploying acoustic receiver with some of the traps as well, which records the vocalization of the lionfish. And then you can pair the videos with those recordings to associate sounds with behavior, so we can get an idea of, do lionfish make some sort of aggregating call that brings other lionfish in that could be useful as a lure in the future? And also, if we can just listen for lionfish. Nope. So if we hear lionfish in the area, that tells us lionfish were present. Sometimes you can't see them, even if we have cameras down or an ROV down, but we can use those acoustic receivers to listen for presence or absence of lionfish and know if we were targeting areas with those lionfish. This was a courtship-associated call that was identified a couple years ago. And that call happens in the evening or sort of nighttime, so we wouldn't see that on camera, but we can hear them calling and kind of be able to determine if lionfish are around. Those courtship-associated calls are also really useful at spawning aggregation sites of grouper and other vocal species. So this is 3 different calls actually of the Nassau Grouper. The first one wasn't a courtship call, the last 2 were. And we've embraced this technology in the grouper moon research as well. The amazing thing about little caiman is that you can get in the water at these spawning sites, which is just great as a diver. But from a research perspective, there's so many ways to assess the population of Nassau Grouper at that site. And so we can combine those methodologies and ground truth the acoustic research, which is still a growing technology. So acoustic noise can be used to identify species, their presence over time, so temporally, and then their presence in certain locations, so spatially. And so if we can actually see the grouper through our video surveys or our stationary cameras, and then also hear the grouper, we can say for sure, yes, they were there. There was yellowfin, there were Nassau Grouper there, and then maybe we can even estimate abundance. And then that information can get extrapolated out to other spawning aggregation sites that don't have the ability to go in the water with their fish and do all of the other research that we do. So if they can only listen for them, but they can't see them, then they can get a better estimate of what's happening at those sites. So some of the, one great project that came out of the acoustic research in Little Cayman was recordings in 2015 through 2017. And we really saw that there was both differences in the call frequency, the call types of Nassau Grouper compared to other grouper at the site. And then you saw differences in when they were calling during a peak spawning day. And so I don't know if you noticed, because this does go pretty quickly, but if you keep an eye on that, when you look at 6 p.m., which I'll try to catch, it gets really quiet around 6:15, and that's when the Nassau Grouper spawn on the site in Little Cayman. And so at 6:15, so it's going to be 18:15 up there, it goes really quiet, and we're still a little uncertain if they go quiet or they just move away from this receiver array. But either way, they go really quiet, and then they get really chatty, really excited about what they did. You know, they're like, "That was great, awesome." So that's really cool, because then we can look at that in different areas, and we can also look at that over time to see, do the grouper continue to spawn at the same time, or is that changing as the population gets bigger or with temperature change? So being able to understand these temporal differences is really cool, and then also spatial differences. So in 2020, the Nassau Grouper, the whole aggregation site kind of moved north about a half a mile and then ended up spawning up here. In 2015 or 17, from this data, it was believed that the yellowfin spawned about in the northern area because of the calls that we heard. So in 2020, we were able to confirm that, because we actually saw the yellowfin spawning with the Nassau Grouper, because the 2 species combined. So by kind of combining those research technologies, you can get a really better picture of what's happening at those spawning sites. So just to wrap it up, we're actually trying to take the data that we're using at Grouper Moon and combine it with a collaboration of acoustic researchers throughout the region, and kind of create this underwater noise data set that will help improve spawning management throughout the region. So that's sort of something hopefully we'll be talking about with you next year, if we can get that funded. And with that, I'm going to leave you to listen to Scott Heppell, who's going to tell you all the other cool stuff we're doing on the Grouper Moon site.
Great, thanks, Alli, for all of that, and I will take that from you. So this is obviously a very broad topic. I'm pretty excited to talk about it, though, and as Alli mentioned and during the introduction, we've been doing a lot of different things obviously at a lot of what we might consider different scales. And apparently I'm going to go the other direction on the, so one of the big questions that we have when trying to put forth a conservation project to rebuild a population is getting more of the individuals of whatever species. And these fish, as you just saw on Nassau Grouper and the Grouper Moon Project, spawn at the west end of Little Cayman Island, and eggs and sperm, fertilization, miracle of life, all that kind of stuff. And then sometime later, little baby Nassau Grouper do or do not show up on the reef. And we have this question about, well, where do those Nassau Grouper come from? Were they locally spawned? Did they come from far away? And we've used some technological equipment, some high technology to answer these questions. And so really asking where do these Nassau Grouper come from? And I'm a biologist and I have a child of my own, so I understand the basics of where babies come from. But perhaps really trying to answer the question is, are these spawning Nassau Grouper leading to more Nassau Grouper on the reef? Is really important from a conservation perspective. And so here is some video of what it looks like when we're underwater, when the Nassau Grouper are spawning, rise up in the water column, and release their gametes, fertilization happens, off they go. And the problem is that once this happens, we're dealing with, something that's really hard to track, because Nassau Grouper eggs are somewhere between 0.8 and 1 millimeter in diameter, and they're basically clear. And so it'd be hard to follow. And because of that, we have worked towards trying to understand the oceanographic processes that tell us where those eggs might be transported, because the eggs can't swim around by themselves. And from that, be able to understand where all those offspring are going. And one of the ways we do this is something called a surface velocity profile drifter, an SVP drifter. And there's a schematic of it here on the left, and for scale, a diver there on the right. And basically here is some images. Probably don't need that volume, sorry. Stop the video, maybe it'd be the easiest to do. There we go, sorry about that. On the left here, 3 pictures of it on the deck of a boat being deployed and floating around. And the basic features of it are that long sock that we see hanging down there that gets pushed around by ocean currents, a tether to the surface, and a large black ball, which contains a big battery pack, a GPS, and a satellite phone. And so I can literally throw this into the ocean, really anywhere in the world, and we're doing it here on a Nassau Grouper aggregation, and tell it every 10 minutes, regardless of where it ends up, just send me a text message with your location. I can get that on my phone. I can track where this is going. And so we let these go on the nights that we see the fish spawning, and use these to track the ocean currents to try to figure out where those eggs might be going. Do they end up on Little Cayman? Do they go to Cuba, Jamaica? Do they go out into the middle of the ocean, never to be seen from again? And what we found, and this is really fascinating from a couple different perspectives, is, and those yellow lines there are what happens when we let those drifters go the nights before they spawn, the blue lines after they spawn. And now what is being shown is that on the nights that the fish spawn, those drifters do something fundamentally different. They actually circulate, they move slower, and they stay closer to the shoreline, which means that those Nassau Grouper on that location are spawning eggs that are potentially recruiting back to the island. And that's really important to justify, in this case, to the Cayman Islands, that conservation of their Nassau Grouper spawning aggregation gives them more fish. Because if those were being carried off somewhere far away, then like, I don't care if they're seeding Cuba or Jamaica or anywhere else. But if they're seeding our own fish, and that is the future of our population, that's really important. I will point out, however, that a big oceanographic drifter like that is not, in fact, a larval fish. So how can this thing that's 5 times taller than a scuba diver underwater actually tell us anything about biology? Well, we'll couple this then with some new stuff. Some of you probably are familiar with plankton nets. Basically, it's a small hoop net that you drag through the water to catch really, really, really tiny things, plankton that floats around. Now, we could just tow this around and then pick it up and look at it and see what was in there and see if there were any eggs in there or not. But of course, that would be too simple. And even though this is a technology that's been around for many, 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 many years, we now have the ability to, in fact, not just tow a plankton net, but through work with engineers at Scripps and elsewhere, design an actual underwater towable microscope. So instead of using that net to collect stuff and then haul it back to the surface, we have this underwater microscope being towed along, and we can actually use it with super cool artificial intelligence, basically machine learning technology, so that anytime an object passes by the microscope while it's being towed underwater, and you can see an image of it down there, it should give us an indication of, tell us, did you see a fish egg or not? And so what we're seeing now playing over here on the right is those 3 green dots are drifters that have been released, and that moving pattern up and down is a boat towing that net. And every time it detects something that is approximately 1 millimeter in diameter and spherical, it says, here's an object. And what you can see is that pretty much every time that boat passes by where the drifters are going, you're seeing a red dot appear. The size of the red dot tells us how many eggs it's detecting. And so it turns out that, in fact, with this super cool optical underwater microscope that we can tow around, we can see these eggs track all around. And it therefore indicates that we can use these drifters as a proxy for the eggs that are moving on. Now, there's some problems with this, and so adapting new technology is always part of what we're doing. It's really hard to control this particular camera, particularly the depth, and as the eggs move up and down to the water column. And so we developed a better optical underwater camera, and this time with wings, so it can actually be flown up and down through the water column, so we can check what are the fish egg concentrations like at 10 meters, at 5 meters, at the surface, up and down. And in doing that, we've loaded up, we've got the device sitting there, this optical net cam, our drifters to track where things are going. And this is just to portray the real glamour of science. It's really exciting. You get stuff loaded on your boat, you head out to sea, you get your equipment out, you get it in the water. And then I should point out that this is, again, the real glamorous part is you track this for 36 hours, towing this thing around and up and down and up and down, and it gets dark, and you find anywhere you can on the boat to sleep, whether it's in a beanbag chair or so on and so forth. But this is a track then of what a 36-hour period of this thing flying up and down in the water column. So that scale there on the right is 10 meters. And so we can actually track it up and down. And these are the things it sees. It sees all sorts of cool plankton-y things that I have no idea what they are, but shown there in the yellow boxes, the machine learning part of the microscope is saying fish egg, fish egg, fish egg every time it sees something like that. And so here's then coming back the next year with this fancier thing and being towed along, and sure enough, we're tracking those drifters and back and forth and back and forth. And so we can use this information and the ocean currents to understand where all of those eggs are going. And now I'm going to step back and tell you that in 2011, it turns out, there was a really, really big spawning event where there were lots of babies that showed up on the reef. And it takes about 6 or 7 years for us to see those in the adult population out where they're spawning. And we saw this big pulse come in. And so with our understanding now of these oceanographic currents and the fact that our drifters can predict where they're going, we can then answer that question, where do the grouper come from, using mathematical models of the ocean patterns and the larval concentrations around the drifters at that time. And this is, I think, super cool, is that if you look at the patterns of where those drifters went in 2011, after 5 days at sea in this big open ocean, 5 days later, they're right back, basically along the shore of Little Cayman. And this was really important to the Cayman government and their Department of Environment to justify that our Nassau Grouper come from our Nassau Grouper spawning aggregation. If we want Nassau Grouper, we should consider conserving this Nassau Grouper aggregation. And that led to changes in their conservation law. The last thing I'm going to talk about very briefly here is actually kind of like what I just talked about, a marriage of very old and high tech and where we're trying to go next. As Alli pointed out, sometimes when we don't have a perfect system like this to work in, we need to be able to detect things acoustically or by other ways when we can't get in and see things. So what I'm holding here is something called a Nansen Bottle. It's a water sampler designed back in the late 1800s. Basically, you can send it down, and then you send on a line, and then you send a heavy weight down. It hits a trigger on the bottle, it closes the end, and so you can take a water sample from whatever depth you want. So why are we water sampling for Nassau Grouper? Well, like everything, Nassau Grouper have DNA in there. They have a whole genome, right? All the genes that make up all life on Earth. Well, it turns out, just like a lot of different things, they shed mucus and scales, and they excrete, and so on and so forth. And so that DNA is not just contained within them, but it is shed into the environment as what we call environmental DNA. And with modern techniques for DNA analysis, we can actually now potentially take a water sample and, without ever seeing anything, tell what's there. Was it a Nassau Grouper? Was it a yellowfin grouper? Maybe it was a French grunt. If you understand the genomes of those animals, you can actually tell what's there, just like a fingerprint or a hair left at a crime scene. You don't have to know who did it, but you can tell that that person, in this case, that fish, was there. Furthermore, if they're not there, we can't see them, we can tell if they're there. But the other important part about it is that if there's one or a few fish, there's a little bit of DNA. If there's several fish, there's lots of DNA. And in fact, we could probably tell the difference between those Nassau and that black grouper there in the background. And if there's lots of fish, there's lots of DNA. And so we have just started now working with this aggregation and water sampling, and we'll be working with the Oregon State Genetics Lab to identify and be able to track how many Nassau Grouper are there, not by looking at them or anything like that, but by actually just water sampling their environment. So that's where we're headed next. I believe Brice is up next.
Okay, awesome. Thanks, Scott. How many people dove today? How many people saw a Nassau Grouper? Yeah? I've heard they're coming back here. I've heard that people are that big. That sounds like a Goliath grouper. That's awesome. Very cool. Yeah, I've heard that people are seeing more and more of these things. So I think things are really looking up for the species, perhaps in no small part to that Central Caribbean aggregation that's now spewing forth great heaving gobs of gametes. Go REEF. All right, so we're going to talk about imaging and AI next, and I'm going to continue along our timeline. So I started this very sort of stacked talk by going back in time to 1993 and showing you our sweet computers from that time that were burning up the internet that didn't exist yet. Right, so let's fast forward about 10 years. Let's fast forward to 2004, and this is really when the Grouper Moon program got started. It started in earnest. This was, in fact, the first year that both Scott and I were at the Grouper Moon project. And not to say that it wasn't already a thing, but we really started to bring in external science and technology to support the program itself. So 2004, what, technologically speaking, was 2004 like? That's a while ago. You probably sort of remember it, but kind of don't. Who remembers Motorola Razors? Yeah, you guys had those? They were sweet, right? You felt really good. You felt good when you had that razor and you flipped it open. It was a comfortable feeling, right? Pop that sucker open. Hello, hello, I'm cool. Yeah, that's me, right? So Motorola, it was still years until the iPhone comes along. So this was modern technology, and all kinds of smaller and smaller flip phones happened. But this was the best-selling model in 2004. What else happened in 2004? Believe it or not, Facebook launched. Mark Zuckerberg at Harvard University, then dropped out, first started Facebook as a community resource for Harvard students, and then now it is a giant spying conglomerate. You may like it. It's not good for you, let's be honest. But that was the start of it, right? So 2004 doesn't sound very far in the distant past when you just say it out loud, but when you think about it in terms of technology, holy cow, have things changed in that amount of time. In 2004, we were using some of the best prosumer grade technology that existed out there to collect images of fish, Nassau Grouper. This Sony Digital 8 Handycam with a whopping 700 times optical zoom. That I don't even think is possible. They're making that up. 700 times, come on, you're snowing me. So that was the camera we were using in that giant housing that took not high definition images, but reasonable, right? Reasonable images. And on top of that, we had 2 lasers. Now, that's technology, right? The moment you throw lasers underwater, we're talking technology. And we had 2 of them. 2 of them mounted on either side of that camera, perfectly parallel. Why? Because no matter where those dots hit something, no matter how far that thing they hit were from me or the camera operator, they were always exactly 20 centimeters apart. Why is that important? If they're 20 centimeters apart, no matter how far away I get them on a fish, I can use that 20 centimeters as a measure to ultimately estimate the size of the fish. Christy's going to talk a little bit more about this later, but 2 things that are most important in fisheries. How many fish are there and how big are they? That allows you to get to biomass. Those 2 things are important. It also allows you to think, get at things like Scott was talking about recruitment, understanding how changes in size frequencies of fish at the aggregation site will reflect whether or not there are new teenagers showing up at the aggregation site. So these are important pieces of information. And back then, this was the cutting edge technology. It required a battery and tape. Imagine that. You kids don't even know what tape is. But it's tape. Cameras used to have tape. It's true. It's true. All right. Let's jump ahead again. Another 10 years, more or less. 2016. You guys remember 2016? It seems like yesterday, right? Yesterday. It wasn't. It was a while ago. Virtual reality was just becoming commercialized. So you had this guy who really is not selling virtual reality. I gotta be honest. I'm not looking at that guy and going, I wanna be him, right? You probably aren't either. It's an interesting Choice Time magazine. Interesting. Nonetheless, this guy's got this virtual reality thing on his face, and it allows him to immerse himself into a world where he is muscular and probably has girlfriends. Let's just assume that's why he's happy. We don't know. We don't know. But in any case, he's immersed himself in, and why? Why is it possible? It's because that binocular vision is giving him depth of perception. So we've moved from a world of 2-dimensional imagery to 3-dimensional imagery. Right? And that allows for a more immersive experience and allows for lots of things that we as scientists can leverage as well. And so we did our own version of virtual reality, and we moved away from lasers into the world of stereo video. This was our first stereo video rig. Imagine carrying that thing around with you. It's huge, right? Huge. But this stereo video rig has a couple of different cameras in the center that are slightly angled inward, just like your eyes are. And your eyes, because you have 2 of them and they cross vision in front of you, allows you to estimate depth. You can estimate how far away I am from you. We can do the same thing using that imagery in post-processing with computers using this system. This worked. The reason, see those black things at the top there? Those are all floats. Why? Because this thing was so darn heavy that we had to rig it up with about 50 floats to make it so that we just wouldn't drop down to the deep briny blue, immediately falling off the back of the boat. Nowadays, and I encourage you to look over there at those TV monitors rather than here, because the big lights up top are blanking this out. It's much more interesting if you look at the nice bright color TV monitors. Nowadays, you're seeing Dr. Scott Heppell and Alli Candelmo. Scott's holding on to our modern stereo video system, which is just 2 GoPros on a very long arm, which we can then post-process using AI technology and computing technology to get estimates of fish. The value of stereo video is now I don't have to do 1 laser at a time on each fish. I can measure all the fish in frame. In this case, what Scott is doing and Alli is doing, Scott is imaging fish that are spawning, that are actually releasing gametes. Alli is then collecting those gametes. And we can take all of that information back into the computer world, and we can say what is the size frequency of fish that are participating in that spawning? What is the big female? What is the size that she is in the front of that leading spawning rush? And then using those eggs and eDNA or DNA, in fact, in this case, we can look at how many adults are participating in fertilizing those eggs, all of which is possible with the technology that we're talking about today. Pretty picture, pretty picture, pretty picture. It's amazing, right? Amazing, the fish spawning there. So we can take the images from this the stereo system that Scott's holding on the left and bring it into an AI framework where the computer software itself recognizes images, individual fish within frame, and then measures those and spits out a spreadsheet basically of fish sizes. Okay, really cool stuff. And it allows us to do something like get these length frequency distributions. So this is basically a distribution of the size of fishes where on the x-axis is millimeters and on the y-axis is year. And I want you to know, Scott mentioned in 2001 there was a big spawning event. Those fish recruited to the reef in 2011. Those fish recruited in 2012 at that size. A bunch of them started getting caught by fly fishermen, and we started getting reports, hey, there's baby Nassau Grouper. There's baby Nassau Grouper that year. Fast forward about 6 years to 2017, and what do you notice about that size frequency distribution in 2017 from our laser and stereo video? You see the multiple humps, right? That's that second hump to the left, that's the teenagers. They're showing up from the first time. They don't know what's going on, but they're ready to go. They're ready for action. And then that year, I am not kidding, we saw young male Nassau Grouper spawning on top of big black grouper. Black grouper's kind of swimming along, and the young ones are like, oh, all over the black grouper, and the black grouper's like, oh my God, I'm not even a Nassau Grouper, get off me. So using this information, it's very powerful to allow us to say, yes, in fact, there was a big recruitment, meaning arrival of new individuals into a population, a big event of that is driving recovery. And it shows up in our estimates of abundance through time using other techniques like marker capture. And so this is the actual population. Y-axis is number of fish from 2008 to 2018. The details of this graph are not so important. Just squint and you will see the lines going up exponentially. That's a good thing. And right around 2017, you see a big jump. That's the recruitment. That's the teenager showing up at the spawning site for the first time. Powerful information that we can then say, look, conservation success is happening. It's working. You guys are conserving this population and you are getting more fish, and those fish are recruiting. They're coming into the population, which all helps us to say something about how to protect these spawning populations into the future. Okay, one more. Fast forward to 2021. This year, modern technology. What's happening? The world is getting taken over by killer robots, basically. Robots are now smarter than us. Artificial intelligence exists, and I don't even know why we're even thinking anymore. They should just be feeding us, and then we should just be out to pasture. Robots, terrifying. So let's sit back and watch a docile video. Again, feast your eyes on the better screens than this one. Here to soothe your terrified soul, thinking about robots taking over the world, is Christy doing a wonderful soothing dive at the aggregation site, looking just like a tourist might do, holding a GoPro selfie stick and shooting pictures of fish. That doesn't look like science. She told us all along she was doing science. In the Cayman Islands, she's not doing science. She's playing with fish. No fair. No fair. We all want to do that, right? Okay. She's not. Actually, she is doing science. She's not playing with fish. Well, she is playing with fish and science. You can do both. It turns out that we can use artificial intelligence, just like your phone does, to recognize faces, not just of people, but in this case, we can recognize the faces of fish. Different Nassau Grouper look different, and they look different consistently through time. And so we can actually, using artificial intelligence and an image workflow, we take those video files that Christy collects, or our DOP cameras, and we push them through a workflow, a computer workflow that we've developed, which automatically slices up that video, segments those videos into fish boxes, identifies those boxes within frame, labels those things, and then we can push that through an identification software. And then by doing that, we can capture images of fish named Bob or Sarah or Judy. We can keep track of individuals in a population through time. This tool is now coming to you all. We've created, at this point, a grouperspotter.org. And this website now, from now on, starting with Nassau Grouper, but soon to include all the grouper you might see, you can take a picture of a Nassau Grouper, submit it into this website, and it will forever be part of a database of individually recognized Nassau Grouper, which will get ultimately matched with other grouper in our database. And that matching of individuals allows us to keep track of individuals through time. Because you all can do that, we can get many eyes underwater collecting that information, which will be very, very powerful to say something about, okay, in the Keys, people are saying Nassau Grouper are coming back. Are they? Are they? There's very little information on them, even with reef data, because they're seen so infrequently. But if we can keep track of individuals, now we're talking. Now we can start to say something. Okay, so here are some of the outputs of that. Take a look at this fish, top and bottom. Is that the same fish? What do you think? The computer says yes. You think it is? No? Show of hands, yes. Put them down. Show of hands, no. Okay, this is why the computer is smarter than us, my friends. This is why the robots are going to blow up our world. This is the same fish, and the computer knows it. It knows it. And if you look carefully, you can see it. You see those dots at the top? If you look at the patterning of dots, it almost looks like the Southern Cross. On the top, you can see it better on the lower one. That pattern of dots remains the same. And if you look across the entire patterning of that fish, if you study it, it becomes apparent that is in fact the same fish. Different months, same fish. Here's one where the computer is not so sure. It says maybe, I don't think so, but maybe. Do you think this is the same fish? I don't either. I don't either. I can see some spots, I could see where it might think maybe, but if you look at the spots, they're just a little off, right? So probably not the same. Probably not the same. How about this one? Same fish? Absolutely, right? Look at those dots. Unmistakable. Unmistakable. Same fish. See the two laser dots on that? We have measurements of this fish. Not only are we keeping track of this fish through time, but we're getting length measurements of that fish through time. So we can get at things like growth. Not only things like survival of Bob, let's call him Bob, but we can get at his growth through time as well. Here's one, we're calling him Waldo. We've seen him 2 years apart now. We saw him in 2019, and then he came back again to spawn in 2020. Same fish, and you can tell based on those spots, right? Pretty clearly. Computer told us the computer doesn't need us. The computer's like, yeah, that's the same fish, but it's the same fish. So we can keep track of these things, and they maintain their patterns over multiple years, and that gives us a very powerful set of information to do things like keep track of ontogenetic shifts. Where does the fish live? Where's its habitat? Does it move between dive sites over time? Does it move deeper over time? Does it get bigger over time? We can estimate growth. This bottom graph there is a growth curve through time. We can also estimate whether or not it goes away. If we're consistently seeing Waldo year over year over year, and all of a sudden Waldo's no longer coming back, that's pretty good evidence to suggest that Waldo has passed from this mortal coil. Or maybe on the menu. Yes, don't eat Waldo, my friends. All right, so very, very powerful information that we're just starting to get a better handle and grasp on. Okay, so I'm going to stop there and let this whole thing get finished up by Christy.
Great. All right. So some really fun technological tools. The one that I'm going to close us out with today kind of goes back to that the stereo video that we were looking at earlier. And well, I'll go back to our acronym, SMILE, Size Matters, Innovative Length Estimates. The SMILE project is something that we started thinking about a year and a half ago. The Fisheries Management Councils approached REEF wanting to leverage our citizen science program to collect data that they weren't quite sure. They just figured that we could capitalize on our big volunteer core of surveyors who are doing surveys and capture more information from them. And what we came up with was this idea of the SMILE project. So Brice talked about how, we're in 2021 now, and kind of the robots are all around us, drones in particular, drones using AI, whether it's the drone up on the Mars rover or surveying a golf course. Those drones need a way to be able to sense the environment and capture and read data from flying around. One of the leaders in creating that technology to be able to sense the environment is Intel. Intel has been creating what they call the real sense technology, where it's capturing depth of field. So it's not just capturing the image, but depth of field and the information about the field that it's taking the video or the photo from. So we've been partnering with Intel and some other folks to turn our surveyors into basically a reef drone, capitalize on all of our surveyors who are out in the water doing fish surveys, and can we add on some piece of technology that will kind of turn us all into drones, and in a good way. So the idea is we're going to take these cameras. This is an idea. You can see these are some prototypes that the divers are diving in an aquarium tank at Birch Aquarium. We've been collaborating with folks from Scripps Institution of Oceanography and the Rades School of Engineering at UCSD and Intel to create this little machine, relatively little, working on getting it smaller and easier. It would be something that we could carry along with our slate. It's not going to be something that would be widely used, but we could have, field teams go out and use it. Maybe we'd have cameras that could get checked out so that people could use them on special projects. We'll probably have them on our reef trips. The idea is we'll have Smile cameras along with our slate, and either we're capturing images of all of the species we see, but probably it's more, especially here in Florida, some of the species that are of most of interest: snapper, grouper, grunts, parrotfish. Now, why is that important? Well, so you can see, as Brice showed a little bit, this is really some cool stuff. These are from that time when they were in the aquarium tank. They were testing it out. These are some actual images from that test. The picture on the upper left is just a regular, RGB photo. That's showing you the fish. And then the one on the right is that same snap, but with the depth of field, the size. So then from that, the AI in the onboard computer is calculating size, girth, length over time. And then one step further, it's also able to start to identify different individual species or fish to species. So it's knowing that, giant sea bass, Garibaldi, in this instance, this was in California. So being able to capitalize on us as our citizen science program to capture an additional layer of information from our surveys. So why do that? Well, as Brice said at the beginning of the talk, abundance is really important. And that's something that we get from the reef data pretty well. But size, we don't have a size measure. For those of you reef surveyors in the audience, we're not capturing size. But if we had something that was able to kind of almost act as a spear, but with our camera, we're spearing capturing images of parrotfish as they swim by, or grouper, snapper, to be able to capture not just the size, but the girth, the healthiness of those different species. Obviously, that kind of data does not exist for most species, especially if they're not in the fishery. So parrotfish, damselfish, Goliath grouper that aren't traditionally caught. So Smile, hopefully, this is kind of our next, like Alli was saying, hopefully the sound project we'll be talking more about next year. Smile will be something that by next year we'll really have on the ground and going, but it will be able to allow us all as kind of our natural historians tapping into reef surveys to be able to collect an additional stream of data that will be really useful for the resource managers that we partner with. So that's kind of on the next horizon. The 2021 robots are taking over the world. Hopefully we're going to be able to use that to our advantage and kind of bring up the Volunteer Fish Survey project. So that brings us to the end. I know we covered a lot, and I really hope that you guys all enjoyed learning a little bit more about our technology and the advances that we've been doing. I know some folks might have questions, so we're happy to take some questions. But before we do that, I'm going to say a couple of announcements just so everybody's on the same page. So after we're done with questions, we've got our happy hour outside or in the lobby. We've got snacks and the bar will be open. Don't miss the silent auction, and a lot of our board and all of our staff are here. And then we'll come back in here at 6 o'clock, which is in about an hour and 10 minutes, hour and 20 minutes, to hear our great talk with Ned and Anna. So those are our quick little announcements. It looks like Jana's got some questions coming in from the live feed. So
We
go ahead.
do. Yeah, we have a question from Facebook from Ofer. And he asks about what Brice was talking about with the recognition work. Wondered if it would work on terminal phase parrotfish, or do individuals vary too much over time? So would that recognition work on parrotfish?
In general, yes, I think it would work on parrotfish. Some more than others. I was looking at a midnight parrotfish today, and I had the exact same thought. I'm like, in fact, I took some pictures of it because I'm pretty certain in the near future we're going to be doing those big parrotfish, the rainbows, blues, and midnights, because they have very characteristic patterning on their faces that I'm certain persists. And I think we're not the only ones doing this individual pattern instance recognition. That's what this is called. This is something that other people are doing it with zebras, with cheetahs, with lions, lots of different, in fact, even with sharks. And the general consensus is that facial recognition, the uniqueness of individuals is far more omnipresent in nature than we give it credit for. And which, of course, if you think about it, makes sense, because, you know, we can all look at each other and we can instantly recognize a person by their face, even at a distance, right? And undoubtedly, most other species are very similar in this way. So the cool thing about the AI techniques that are evolving very, very rapidly is they're getting better and better and better at that pattern instance recognition. And so I suspect that we're going to rapidly move from not just Nassau Grouper, but all grouper species and into other important fish species that we can readily capture information on, to include parrotfishes.
Great, thanks. And for other questions, we have 3 mics floating around the room. So if you can wait for us to give you the mic. Here's one back here. Dun, 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 dun.
Thank you for a fascinating presentation. I am from the Los Angeles Maritime Institute. I'm the education director. Thank you for inviting us. I apologize that we're going to have to leave soon to head back. My question is a little bit off this topic, but related. We in LA have been working with REEF and other organizations very concerned about the impact of microplastics in the marine environment. We don't have any real reefs there to study, but we do a lot of study on microplastics in birds, in fish, etc. I'm wondering if, in what you have seen, any observable data on the increase in ingestion of microplastics in any of the grouper or other fish, because we're seeing it in every single marine organism studied, from single-celled organisms on, and it is part of the devastation of the oceans.
Right. That's a really fascinating question. The short answer for us with our grouper right now is no, because of course the way to answer that question is to do stomach content analysis, and we're not capturing fish. In fact, a lot of this technology that you've seen is really pushing the frontiers of conservation science to get us away from handling critically endangered Nassau grouper and things like that. The long answer to your question is almost certainly yes. And there's a lot of work going on the West Coast now, up and down the coast, trying to answer those questions. For these particular species, we don't know.
What about hormone disruption, intersex fish? I know they've seen those in rivers in West Virginia and probably other places. Anything like that going on?
As it relates to plastic or just in general? So there's not, based on my reading of the plastics literature, there's very, very little evidence to suggest that plastics confer any amount of hormonal toxins that would do something like sexual disruption. Plastics have lots of different things that are bad, and one of the things that plastics do is they are aggregators of persistent organic pollutants, or POPs. So if you take a piece of microplastic out of the environment and you analyze it for these pollutants, there'll be a lot of them on there. The thing is, when you take those and you feed them to animals in the aquarium, the plastic holds on to the pollutants. The animals don't take it off the plastic. And so there's very little evidence that so far to suggest that there's a huge amount of toxin transfer from plastics to animals. Now, don't take that as me saying that plastics aren't bad. I'm just saying that plastics are not, as far as I'm aware, big in terms of loading biological organisms with toxins.
So I see you concentrating on groupers. Is there a plan in the future to expand to like turtles or any other types of species or anything like that?
I feel like that's a director of REEF question, but let me opine. Absolutely. And in fact, you're going to hear a presentation from Dr. Dan Greenberg tomorrow. Stay tuned. And he's going to be talking about some of the new quantitative tools and techniques that we're developing in order to analyze the data that you all collect. And so because we've got now 30 years worth of data, especially rich data from the Florida Keys, Dan has developed some cutting edge statistical modeling approaches to look at trends and trajectories of animals, not just grouper, but lots of different species of fish. In fact, 100 different species of fish, more or less, a lot from the Florida Keys. So he's going to talk about that. Those tools and techniques that we're working on, collaborating on developing, are applicable not just to fishes, but to turtles. And that's one of our very next big projects that we're hoping to tackle is the turtle data that REEF contains. REEF is unique in that we all are making observations on turtles, and in places where turtles, where wildlife programs don't have nesting beach surveys, there is almost no information on the abundance and change in abundance of turtles through time. Throughout a lot of the Caribbean, for instance, except there's REEF data. And so those REEF data are actually very, very powerful and untapped to look at trends and trajectories of turtle populations. So we're excited to do that. So that's the long answer. The short one is absolutely, it's all coming. Just give us a little bit of time. Doug.
So as REEF and other environmental organizations have embraced these new technologies to really start to consume this information that's available, have the technology companies taken the other side and become very interested in what environmentalists are doing and pursued their new inventions and technology towards you? So you've adopted them. Have they decided that they need to adopt the environmental community as well?
Okay, I'm going to answer this one, and then the next one of you all gets to answer. So Christy pointed out that Intel is actually working directly with us on that new fish sense tool, the SMILE program. And that was one of those things where they didn't come to us, we came to them and we're like, hey, we're using your tools underwater, but we're getting some weird artifacts in terms of our size measurements. And we're like, yeah, well, that's because we didn't design our algorithms to work underwater, but we can. And so now they're working with us to change some of their algorithms to do better measurements of things underwater. So a little bit. They're not exactly dumping money on us, but that's okay. If we were to have developed those real sense cameras from scratch, I mean, you couldn't do it with all the money, research money in the world. I mean, they've got a team of scientists miles deep that have been working on this problem for decades. So we get to reap the benefits of that. And all it takes is a little bit of creativity on our part and some willingness to buy in on their part. Yeah.
So I thought the thing you mentioned about us being able to upload our photos from surveys was really exciting. And I think you said it was live now. Can you remind us how we do that?
So the website is live, but we're working out some kinks. And we've just been figuring out how best to enter all the metadata. And the company that we're working with, WildMe, is also a nonprofit. So we're hammering them with questions on a regular basis. And we uploaded about 10,000 photos all at once, which is probably the most that they have in their entire database. So we overloaded the system. A lot of things didn't work. We've been trying to figure out why. So we're pulling it back, and we're going to start with one bulk upload at a time. get it working and then advertise it out. They're also switching their platform. So we're going to wait until we go into the new platform before we announce it more publicly than we already have on the live feed of Facebook. So I guess the answer is don't upload any photos right now because we're going to delete them all next week. But stay tuned. We'll announce it soon when you're ready for it.
Smart, and the people from Intel could be encouraged by the work that we're doing and what they're getting out of their product. Maybe we could use that to get some monies for REEF. They benefit by what we do. And this is nothing on the topic, but I'd like to address the group. My name is Kathy Comerford. I'm one of the original people in REEF. And I was at Matangi, Fiji 5 years ago for my 70th birthday. This is my 75th. And I was fortunate enough to be able to do my work with my husband for 10 days. And we did the baseline survey there. They are now having a huge problem. One of the owners of a resort in Fiji wants to reshape the reef so it will become a surfing area and he can make money out of it. And he wants people who are the fishermen and support their families and eat from their permits, give them $25 US for that permit. And my girlfriend, who is the owner of Matangi Island, her great-grandmother was a Fijian princess, and they've been very good stewards of their island, are trying to fight this. They've had many meetings with people, the fishing people and the fishing community of Fiji. And I would really appreciate if anybody's got data on trips to Fiji, any of the areas, and pictures. I am at Toucans, TOUCANS, @me.com. Forward it to me. I am going to get it to Christy. Mr. Cousteau has already written a letter, which I've given to Christy, and we need support for this. It's very, very sad. Their reef, their private reef, their marine preserve at Matangi, 90 feet right to the bottom, clear during cyclone season. I saw white tips, black tips, and the most gorgeous colors of any reef I've ever seen in the world. It was pretty impressive. So we need to save places like this. So hopefully someday you'll be able to visit, or your grandchildren. Thank you.
Yeah, thanks, Kathy. Yeah, that's a crazy situation there. So hopefully we'll be able to get them some of our reef data and yeah, photos. And so, any other questions? All right, well, we'll obviously be around out there, so feel free to grab any of us. We really appreciate y'all being here today. Enjoy the happy hour and then come back at 6.
keys. Go, there's no place left.
Hey, we're still live, Martha.
Oh, hi, hello, how's it going?
You guys all know Martha. So we're doing an impromptu after the seminar, which went very well.
It went great. Thanks so much for joining in. For those of you online, I know a lot of you couldn't be here, and we appreciate you being careful because of COVID precautions. And we hope that next year it'll work out for more of you at home to join with us here in person. But thanks for joining in online.
Thank you, Martha, the executive director of REEF. We're checking out happy hour here. It's a great turnout. Great time, Martha. One of our speakers, Brice. Hey, nice spread. All these pics up here are Stephen Frank, world-famous photographer from Key Largo. The county building's now closed. It's Friday afternoon, so now it's ours. The Reef Fest Happy Hour. We have new shirts this year. All right. Check out what's going on.
He's here.
David, we're still live.
Hey, David, how are we doing?
We've been asking to talk to you.
Oh,
How's it been going here on the outside?
on the outside? For
Out here.
oh, right on. It's been going great. We're all set up for Reef Fest this year. We encourage everybody to stop by. You can stop by, check out our silent auction, our raffle. Come by, get an iguana bait, get a red or white wine. Definitely check us out. Promote Reef and promote marine conservation.
Excellent.
Yeah.
What's going on over here?
What's going over here? We got our well, first of all, that's Andrew with his
Hey,
iguana
Andrew.
bait. Shout out Florida Keys
Well,
Brewing.
more important, what's going on back here?
Sure. I will take one. Thank you so much.
You have to open one online, right?
Excellent. Yeah, I got to open it up.
Post
Florida
the mic.
Keys. Oh, that's sorry.
Okay, that's what the filter's
That's
for.
what the filter's for. Okay.
Hey, where's the interns? Well, we're all here. We're all here for the interns. Thanks, guys. We're still live online, so we're checking with our audience who just watched the seminars. We're live on REEF Facebook page and REEF YouTube. I would, yes, please. So this is Lauren, and Lauren. And Haley. All right, where's the other Lauren? That's yours. The other Lauren. You said interns. You have interns every semester. You have a beard. Oh, I got a glass too. Fancy smancy. Oh, yeah. That's Lauren we were hanging out yesterday. Lauren from the Netherlands, right? Yeah. Yeah, I remember. She's from The Hague. Yeah. We hung out yesterday at the Reef Social.
There you go.
At the open house in there. It's Reef Beer. Thank you, guys. This is a big part of Reef every year is a silent auction. It's how they raise their money for this event. Who knows? Come on, check out the items. Right here, pardon me. You're hanging out at Reef Fest Happy Hour at Murray Nelson Center, Hallmark Awards in Key Largo. You're just catching this year around, come on down. Ned and Deloach, co-founders with Reef, are speaking tonight at 6. Lots of cool items.
I tried to call Leo. Yeah, is he still here? I'm going to stay live. The point is on experience. I'm not a leader. I have to take the audience.
Here's right here. Hey, Daryl, good to see you. That was Daryl, my assistant. David Hardman here, hanging out at Reef Fest. Hey, wave, guys. There's our speakers from the last seminar, Conservation Stories. Hey! Oh my God, say hi to me. We're live. You know, I just thought of you to the beneath the sea, but we'll catch up. This girl caught a 13-pound lobster in North Carolina. Weird, I'll tell you why in a second why I came out. See, that's why you come to Reef Fest, see old friends. Here's the hangout area. So the seminar is empty. We're getting ready for Ned and Ann Deloach, 6 p.m. We'll be streaming live here on Reef Facebook and Reef Speak Fish YouTube channel. Please subscribe to We Speak Fish YouTube channel and like this page. And as always, like and share if you watch it. It helps us out. We're going to get food soon. It's going quick. Everyone wave. Gianna did a great job today. Appreciate you guys. There's Leo. Hi, Summer. How are you?
Good.
We're still live.
Yeah.
Summer was a big help when I was getting this set up initially for the feed. I had to get the social accounts from Reef into my streaming software, Streamer I'm using now. If it wasn't for her, it wouldn't have happened because Stacy's channel is so far locked down on Facebook.
Yeah.
You did, thank you. Trying to talk through the glass. I'm just happy to see this desk being used. It's never used. The county never uses it. It's a beautiful desk here the county never uses. So this is our growth management. In other words, zoning. You don't ever want to go there. Upstairs is Holly Rasheen's office, our county commissioner, some other offices. It's not our main government building. That's at mile marker 88. This is mile marker 102, right across from where I live, right on the bayside on the ocean side. Oh, Ashley's out there. There's the man Leo. He's a big help. Wait, Leo. Say hello.
Hello, everybody.
So Leo got a new shirt. Is that a nice surprise for you?
Oh, yes. They surprised me with that.
That was all Daryl. Daryl set that up. Daryl with the help of Stacy.
They told me they don't have it.
Oh, yeah. I mean, they did for your life. So Daryl works at Public Works, and he stays around all weekend to take care of us every Reef Fest and Ocean Life, too, when it happens. So we appreciate you, Leo.
Thank you.
We're going to check out outside. I can't go too far because I'm on the Wi-Fi coverage. Thankfully, the Wi-Fi is better. I can pull this off here. Pretty soon, I'm going to end this feed. Then we're going to a separate broadcast live at 6. So if this goes out, don't freak out. We already have a scheduled broadcast on the YouTube channel. Hey, gorgeous day out here. I would walk to the park, but I'm going to lose connection. I'll walk out a little bit. Well, nice little social break for everyone watching Reef Fest. Please just stick around today. Shorter seminars today, but 2 of them. When I end this broadcast, if you missed the seminar, it'll be on recording on Facebook and YouTube, where you're watching it here. And this is the county building. Well, what a community room. I've been involved with the Friends at Keeler Agricultural Center for 10 years. We bring events here. It was first built in 06, nothing was coming here. Here's some friends eating. Woo-hoo. So one more walk through the happy hour, and then I'm going to close it down, and then we'll see you guys at 6 for Ned and Deloach, one of the marquee presentations of the whole festival. More Steve Fink photography up top there. Thank you to our sponsors. Hi. And here's the co-founders of Reef. Paul's going to be recording. He's not here today. He had back surgery. So if you're expecting Paul, you'll still see him. But they're infamous for these books. They're in every dive shop worldwide. Numerous different languages and for sale right now. We'll autograph it for you. I got a few autographs. That is our check-in on Reef Happy Hour, guys. We got to get set up. The next presentation, 6 p.m. tonight. Ned and Anna Deloach with a recorded version of Paul Honan. It's going to be awesome, spectacular. Live here, Reef Facebook and We Speak Fish YouTube channel. We'll see you then.